observations.
All right, thank you. So before I get into my slides, I thought I'd just let you take a look at the simulation I'll be talking about. There's way more information going on up there than I really want to tell you right now, but it's sort of fun to watch, so now you can keep that in your mind as I start talking about some of the emergence in our system. As was mentioned, I'm Chris Marriott. I'm from the University of Washington. I'm a lecturer there, and my colleague Jobran Chebib is a biologist who's doing his PhD at the University of Zurich, and we've been collaborating for a year or two on some interesting simulations. So one of the things I want to talk about today, or one of the things we were interested in achieving in our simulation, is multi-level emergence. And that's the idea that we might have emergence happening at various levels in our simulation or our system. So for instance, I've highlighted a few areas here: genetic, individual, population, and social levels, as being different levels in the system where we might see some type of emergent dynamics. All of these emergent dynamics happen in our simulation. I want to contrast that to what I'm sort of calling the standard methodology. Well, I went to the Cosmos workshop this week, and you might call this the Cosmos methodology. You have some emergent system you want to study, say herding behaviors. You identify some underlying system like individual agent behaviors. And then the methodology is, well, you model those underlying system with maybe some simple rules, or what I might call a linear system, something that's pretty easy to set up. And the goal is to hopefully have some kind of emergent behavior on the level of the system that you're interested in. So most of us are probably familiar with Reynolds rules for how to get a flock to form. Simple agent behavior, you get an emergent behavior above. One of the drawbacks we see with this is that typically that underlying system itself is also in the real world an emergent system emerging from something else. So agent behavior is rarely just scripted. It is instead emerging from maybe genetic dynamics or neural dynamics and so on. So our goal was to try and design a system where we have maybe multiple levels where maybe agent behaviors emerge out of something, and then from those agent behaviors we might get some social or population level emergence as well. So I'm going to take the first bit of the talk here to just talk a little bit about the model. We follow this common principle in A-Life, that is, if you want to have some complex behaviors in your agents, you need a complex environment. So we use a random geometric network, as you see here. This one has 100 nodes using a reach of 0.2. And each one of the locations in our network is a resource gathering site that our agents can gather resources from. Each site will either have 1, 2, or 3 resources. And every time you go to a gathering site or an agent goes to a gathering site, they're successful at gathering those resources. The cost, however, how much energy it costs them to gather those resources varies, and that's going to be dependent on a strategy encoded in their genome. Furthermore, our agents have to move around the network, so there's also an energy cost associated to that. So let me talk a little bit more about the genome of our agents, which is going to be important to some of our dynamics. So I've got a sort of simplified geometric network up there with just 5s, just so we can talk about it simply. And this would be a possible genome for our agents, which would be a path through that network. Okay, and you can verify yourself that this is indeed a path. So all of our genomes will be a path in the network. I've also indicated with different lengths here for each site, indicating maybe the energy costs that might accrue to the agent for gathering at that site. So you notice there's 2 locations for site B there, but they're of different sites. That's indicating that maybe the agent uses a different strategy the second time it arrives at site B, and maybe that's a better strategy, so it takes less energy to gather the same amount of resources. Again, one thing that's sort of important is how does that genome then turn into behavior of my agent? So the way we do that is we one of our principles, again, is we want some interaction between the environment and the genome to arrive in a phenotype, and we usually call that the genotype-phenotype map. Our mapping is pretty simplistic, but it still has some interesting properties. Here's how it works. So when the agent wakes up or maybe is born in a particular site, it has to start out its day's worth of gathering from that site. So it searches its genome forwards and backwards to find all locations of the current site that it's in. From that site, it can start tracing out a subpath through its genome, a subpath because it won't have enough energy in the day to follow its entire genome, so it just tries to find a local chunk of that genome that will hopefully maximize its expected resource gain. So it will trace out from that site enough energy that it maybe has. So every agent starts out with 50 energy, but after time it may have lost or gained energy, so it uses its local energy. And then it tries to maximize its resource gain. Once it finds that local subpath that it thinks it will maximize its resource gain, it just carries out those actions during the day following through. Now normally, if the agent's lucky, it will get the resources that it expects. It will go to every site and be successful at gathering the resources. The only outcome that might change this is if it arrives at a site and too many other agents have already gathered at that site, so there are no more resources left. It will still spend its energy on the site, but it will get no resources for that. We call that a depleted site. Another principle we follow is we think that both life and death of our agents, as well as the reproductive success of our agents, should be related to their actions in the environment. So one thing we don't have is we don't have an objective fitness function in our simulation. This has been a trend that I've seen in a number of talks, is sometimes that objective fitness function, one, it's hard to come up with, but also can kind of hamstring your evolution. And so we use things like novelty search and so on. But we're not using novelty search here. We're just using natural selection. The idea that if you run out of energy and die, then you're probably not going to be as successful reproducing. Also, if you can gain enough energy over time to pass a threshold, then that's what's going to trigger your reproduction activity. So we do have this idea that at the end of the day, you take your resources, you trade them in for energy, and that's going to either give you a net energy gain or a net energy loss for that day. If you can carry out the gain for enough time, you'll pass the threshold and maybe reproduce. Carry out a loss for enough time, you will reach zero and die. So how does the reproduction work in our simulation? Well, as is common in many simulations, we have both asexual and sexual reproduction. Again, we sort of follow a principle of conservation of energy. If I'm going to be creating a new agent that comes alive with 50 energy, then we're going to charge the parent agent 50 energy to create that new offspring. What that means is if you're doing it asexually, of course, you have to invest all your energy into that, so a full cost of 50. If you're doing that sexually, you get the benefit of, you get to split that energy cost amongst the two parents. Now, when you are sexually reproducing, of course, there's also the cost of sexual reproduction, which was part of one of the things we were interested in studying, which is that to carry out sexual reproduction, you need two agents in the same place at the same time, willing to carry out that sexual reproduction. This is sometimes called the cost of sexual reproduction, or sometimes also called the encounter problem of sexual reproduction. So that's the two types, and let me talk a little bit more about how the genetics work in reproduction. So because our genome is a path in the network, of course it's going to be important that after the mutation events and so on, that we're still left with a path that's going to be important to our model. So there's sort of two interesting ways that we could change the path for a genome. One is you could just start at one of the two ends and sort of randomly grow off the end. So that's an easier way to do it. Or you could delete off the end as well. But the two that I want to talk about here at the bottom here is where we search through the genome and try and find a cycle. So I've indicated two cycles in the original sequence up there. That's a sequence of states in our path that both begin and end at the same state. We have one that starts and ends at 8, another one that starts and ends at 4. The interesting part of a cycle in a path in a network is, of course, you could delete that cycle and replace it with just the one site, and it will still be a path in the network. Or you can duplicate that cycle and maybe put it somewhere else. So you find another location. So the first alteration I have there is I've just deleted out the yellow sequence, which leaves us still with a path. The second one, we find another location where there was an 8 in our string, and we can replace just that single 8 with a cycle that starts and ends at 8. Okay? These will make sure that we maintain the invariant that after our mutation event, we still have a path in the network. Sexual recombination is also a little bit trickier when you're dealing with a path like this. In a common sexual reproduction, you might have in a common genome, you might have specific loci, each one indicating something specific, maybe a weight in a neural network or something. And it becomes very straightforward to say, well, at that loci, I'm either just going to take the mother's contribution or the father's contribution in a sort of 50-50 chance. Well, that doesn't work out very well for our path. First of all, our genomes can be of different lengths to start out, so just lining them up doesn't make a lot of sense in that sense. But also, each loci, just because it's the first site in your genome, doesn't mean that's going to correspond well to the first site in my genome. So what we do in this case is we try and find the longest common subsequence between our two genomes. And that's indicated in purple there, so I have a father sequence and a mother sequence. So the purple ones are the longest common subsequence of those two genomes. And that's where we line up our genomes. Okay? And it causes there to be little bulging spots. So you can see that sort of long sequence of red in the mother sequence. That doesn't correspond to anything in the father sequence. So the way we carry out our recombination here is we'll move through that longest common subsequence, and each time we reach one of these crossover points, we decide, well, should I keep copying from the mother sequence here, or should I copy from the father sequence? And we'll copy all the way until we reach the next crossover point where we'll do the same thing. So what this can do is obviously create an offspring sequence that has components from the mother and components from the father, and maybe of different length than either of the original two sequences. So now let me mention a little bit about the experiment that we ran. So the actual experiment we were interested in, which we presented at Gecko, some of you may have seen my colleague present that last week, was we were interested in how agents might find each other to carry out sexual reproduction, so solving this sexual reproduction task. And of course, we got lots of interesting results there. I'd love to talk to you about it maybe afterwards. But the purpose of today's talk is to see that because we took this emergent focus at the beginning, we actually got a lot of interesting genetic emergent dynamics out as well. And I've got a short list of them here, but I'll just talk about them in turn. So the first thing, which is often desirable, is to have some kind of divergence between your genotype and your phenotype. So in this case, because our genomes are paths in the network, we can compare two genomes by using the Levenshtein edit distance to see how close or far away they are. Well, also our daily behavior, which is the phenotype in this case, is also a path in the network, the path that you followed today doing your resource gathering. So again, we can use the Levenshtein edit distance to do a comparison there. Now, what we've done in this diagram here is we've taken a final population after one of our simulations. We put all the agents along the x-axis, all the agents along the y-axis in the same order, and then every point in one of these heat maps represents the difference between those two agents, either genotypically or phenotypically. And of course, after you create one of these heat maps, you usually just don't care about the heat map. You want to now cluster it and see which are the similar agents and which are the dissimilar ones. So if you take a look at the top right corner there, we have the clustering of our agents based on their phenotype, so the ones that acted the same during the day. All right? And you can see along the diagonal, if you check out each one of those sort of dark black boxes, those indicate a group of similar agents that have carried out the same actions during that day. We highlighted one in purple there so we can see. Well, if we cluster the agents using that same method, but instead look at their genotype, that's the top left case now, we see that those same agents that were all carrying out the same behavior during the day actually came from different genetic groups. So even though they have the same phenotype, they came from different genotype groups. Now on the bottom level, we see this sort of doing the same comparison, but from the other direction. We've now clustered it according to the genotype, and we see there's sort of three strong genotypic groups, or different species, if you want to call them that. But looking at any one of those species, we see that they have a wide range of phenotypic behavior coming out of that genotype. So we have this divergence from genotype to phenotype as a result of our model. Another important observation that we have is that our agents, they will typically find themselves in a local optimum, which means maybe the first couple days they wander around the network trying to find a good spot, but once they find a good subpath that they like, they will typically repeat it over and again, back and forth, day after day. All right? What that means is one part of their genome gets a lot of use. Okay? And we might make an analogy here to the coding sequences in DNA. And then there's other parts of the genome that don't get any use, and we might call those the non-coding sequences, or maybe they get infrequent use. Well, in biology, there's a lot of different kinds of non-coding sequences. Some actually code for other things or perform important roles in the genome. But the type of non-coding sequence that we think our non-coding regions are most like is called a pseudogene. Now, a pseudogene in biology is a genetic sequence that is homologous to a gene. It looks like a gene because of its sequence, but for whatever reason, it's not expressed in the genome. Okay? Well, that's sort of true of our case. We have these sequences that could be expressed, but for whatever reason, in our case it's environmental contingency, they're not expressed. So we will call them pseudogenes. So we got this emerging as well, this non-coding region. Now it's important for us to recognize this because this has an impact on the type of mutations that we have. So one thing we've noticed is this. If you consider that region of the genome that does not code, that is not expressed, if you take our duplication, duplication mutation, and we copy one cycle from that and we put it somewhere else, that's because we're not using that region, or rather because the parent agent's not using that region, it's most likely that the child agent, the offspring agent, will not use that region either. So that kind of mutation is neutral. It doesn't have an effect on the phenotype of the offspring. As a result, we would expect that over time that these neutral mutations would accumulate in our genome. What we see here on the right is a graph of the length of the genome over time through our simulations. And indeed, we see that the length of our genome tends to grow. And one of the reasons, or one of our reasons we think that happens, is through this neutral drift or growth through our genome. So that's what happens if we get one of those genome duplications happening in the non-coding region. What happens if we get genome duplication happening on the coding region? And this is really an interesting case. So I'm going to specifically hone in on, imagine that whole region that was being used by the parents is completely duplicated and copied somewhere else into the genome. So now the offspring agent is going to have 2 copies of this region. Now, this will still not have an effect on the phenotype of the child agent. It can only use one of those 2 regions. Okay? And even if they tie, it'll just pick one to use. So we won't actually see any change in the phenotype of the child agent, but what I want to express here is there's going to be an interesting change in the evolutionary dynamics of that child agent. And let's consider a couple cases. So the parent agent that only has one of these sequences, if it gets a negative mutation in there, it's stuck with it. The offspring is stuck with that negative mutation. But the offspring that has 2 of those sequences, if it gets a negative mutation in one of them, it's got a backup region that it can go ahead and still use, so its phenotype will not be upset. What that means is it becomes a little bit more robust to that kind of negative mutation. Okay? It needs to have 2 of those negative mutations in both of its sequences before it will suffer phenotypically. So that's sort of reducing the probability of getting a negative mutation. We might call that robustness. Well, imagine now also consider the case of a positive mutation. Well, again, in the parent, if it has a positive mutation in that sequence, the offspring will have a benefit to its phenotype. But when you think of the offspring that has 2 of these sequences, it now has double the chance of getting a positive mutation. Each of the sequences might have the opportunity to have a positive mutation, and we might sometimes characterize that as greater evolvability, the ability to integrate positive mutations into your genome. So we see both of these cases occurring in our simulation. So some of the conclusions I want to come with here is that we sort of wanted to contrast our model against sort of the standard methodology, where we wanted to incorporate as many nonlinear systems into our model as possible so that there was the potential for more emergence. Not only did we get some of the interesting emergent social behaviors that we were looking for, but we got a nice rich batch of interesting emergent genetic behavior as well. So we hope that we can inspire others to try and use a similar type of technique. Where are we going with this? Well, this was a stepping stone in our research to something else we're interested in. So sort of short term, what we're looking at is maybe tracking that genetic drift a little better so we can get a better characterization of it. Likewise, we want to come up with a way to measure evolvability and robustness in our system in maybe a quantitative way so that we can also characterize that. Long term, what we're hoping to do is develop our agents a bit more, add things like learning and social learning in them. Our final goal is to study division of labor and resource management. So to conclude here, I just want to thank our agents that lived and died in this model to bring us these results. And also, if you're interested, you can check out the simulation, which I had playing at the beginning. It's live on the web, or you can check out our code. And at this point, I want to say thank you and open it up to questions.